This slide carries on with the idea that all living things are related to each other. In other words, united by common ancestry. So again, here we see a phylogenetic tree that represents that. This phylogenetic tree is supposed to represent all life on the planet. Obviously, you can't show every single species because there are billions of species, but it represents the major groups of life that have evolved over 4 billion years on the Earth. Another unifying idea is something that you're already familiar with, that all living things have DNA. Likewise, something that you're not as familiar with probably is this process known as aerobic cellular respiration, which is something that we're going to be learning about a lot. It has a lot to do with metabolism and how your body gets energy, and how almost every living thing on the planet gets their energy, their cellular energy, to do what we call cellular work. So both of those are unifying ideas. The fact that every living thing has DNA and most living things use aerobic cellular respiration to get their energy suggests that we're all related to each other. So we're unified by common ancestry. But over the 4 billion years that life has been evolving on this planet, there have been a lot of branches. The reason there are so many species is because there are so many ways to make a living on this planet. You might be familiar with the term niche, right? So the reason there are so many species is because there are so many niches. There are billions of niches, therefore there are billions of species. It's that concept, if you watch the original Jurassic Park movie, Jeff Goldblum's character said, "Life will find a way." And that's the idea. If there's a way to make a living on this planet, life will find a way to make a living. Another big idea in biology is just the whole idea of science as a way of knowing. What we know in biology is what we've learned through science and the scientific method, carrying out scientific experimentation. So this diagram is a little bit different than what we've seen before when it comes to the scientific method. As you know, the scientific method begins with observation. Well, they've replaced that with research. So research, observation, kind of the same thing here. Just being aware of the natural world, studying the natural world, and then finding a problem or having a question, and then hypothesizing to answer that question, and so forth. So, like I said, you're already familiar with this, and in carrying out your research project, this is where you are right now. You're trying to come up, you're observing and researching and trying to come up with an idea, a question to investigate. That's where you are right now, step one. This big idea is not just a big idea, it's a huge idea in biology, structure and function. And the idea is that at every level, meaning starting with molecules. Molecules have a particular structure so that they carry out a particular function in living things, all the way up through even to the way that the biosphere is constructed. The structure of the biosphere affects how it functions. So there's this direct relationship between the structure of things in living systems and their function. For example, the forelimbs, these are all forelimbs of different mammalian species, and you may recognize them as carrying out different functions. For example, a human arm is for picking things up and grasping things and manipulating things, no longer for walking, right? Whereas a horse's hoof is very specialized for walking and running. A cat's forelimb for not only walking and running, but capturing prey. A bat's wing to fly, a bird's wing to fly. Notice the difference, though, between the bat's wing and the bird's wing. So even though they're both for flight, they have evolved a different structure to carry out that same function, and that's because they evolved flight separate from each other. In other words, bats are not related to birds, and birds are not closely related to bats. They evolved the ability to fly independent of one another. And then there's the whale. And you can see that the bone structure inside the whale's flipper is very similar to even what you have in your arm and hand. And that's because of the close relationship between all these species, because they're all mammals, and they all diverged, as we say, from a common ancestor. So this similarity in structure is known as homologous. The similarity in structure that is the result of common ancestry is known as homologous structure. And as I explained, these are all different because they've been modified through evolution to perform different functions. So the differences here are the result of what we call divergence, divergent evolution, species becoming different from each other to live in different niches. This big idea is known as the hierarchy of biological organization, and it's simply how things are put together, how living things are put together and living systems are put together, all the way from what are known as subatomic particles that you may be aware of, that you've learned in other science classes, that make up atoms, like protons, neutrons, and electrons, all the way up through the biosphere that represents all life on the planet. So in other words, it would be very good for you to be able to recite these in order, and I like to go from the small to the big. So in other words, be able to, from memory, say that subatomic particles make up atoms, and atoms make up macromolecules, macro just meaning big molecules, because those tend to be the molecules that make up living things. The molecules, the compounds that make up living things are big macromolecules. Macromolecules make up organelles, which are the organ-like structures inside cells. Up to this point, nothing is considered alive until we get to the level of cells. So organelles make up cells. That's the basic unit of life. That's the smallest thing that can be considered alive. Cells make up tissues that are specialized to carry out particular functions. The tissues come together to make up organs, which also are specialized to perform particular functions. Organs come together into organ systems, like the respiratory system, the circulatory system, nervous system. Those are also specialized to carry out particular functions. Organ systems come together into organisms, in other words, the entire individual. And then individuals or organisms make up populations. In ecosystems, you'll find populations of organisms. The populations come together to form communities. So these are all the species that are living together in an ecosystem. That would be the community of that ecosystem. Communities, again, are part of ecosystems. There's also the non-living part of ecosystems, so keep that in mind too. Communities are only, the community of an ecosystem is only the living part. There's also a non-living part to ecosystems. And then ecosystems come together to form biomes, which are these vast regions on the earth that are determined by climate and soil and the communities that live there. And then biomes come together and form the biosphere, representing all life on the planet. So that's the hierarchy of biological organization, kind of represented here in concentric circles. So this brings us to the tools and procedures that biologists use commonly. And the first one here is not only used by biologists, but all scientists. And that is what's known as the System International, which specifies that the metric system is the unit of measure in science, period. We don't use inches and pounds. We don't use the imperial system at all in science. And I expect you to do the same. I don't expect to see any imperial measures in anything that you do in this course. So the metric system is king here. And that's because it's based on 10. It makes a lot more sense. It's not as arbitrary as the imperial system. The imperial system is like, you know, some king at some point said, okay, a yard's going to be the distance from the tip of my nose to the tip of my finger, or something like that. It's completely arbitrary. So again, it's known as the System International, and it's the universal system, measuring system used in science. The base units for length is the meter, for mass is the gram, for volume is the liter, and for temperature is degrees Celsius. And we use these prefixes. So for example, 1/10 of a meter is a decimeter. We don't use that unit very often, but it exists. 1/100th of a meter is a centimeter. So centa meaning 100th, 0.01, right? 100th. A millimeter is 1,000th of a meter. A micrometer is 1/1,000,000th of a meter, and you may not be that familiar with that either, because now we're getting into the realm of the very small. And but this is the unit of measure under the light microscope. It's also known as the micron. So micrometer and micron mean the same thing. Micron is short for micrometer. And again, that's 1/1,000,000th of a meter. So you can imagine it's very small, and that's the measure of unit that we use primarily under the light microscope. Then there's the nanometer, which is 1/1000,000th of a meter, which is what is used to measure the wavelengths of light. Wavelengths of light are measured in nanometers. 1/1000,000th of a meter. And then the picometer is 1/1,000,000th of a meter. Extremely small, and I wouldn't be at all surprised if you've never heard of a picometer. I don't think I ever heard of it until I got to college. And then the kilogram or kilometer, kilo means 1,000, right? So kilometer or kilometer is 1,000 meters, and a kilogram is 1,000 grams. So I'll be very disappointed if I see you using imperial units on anything that you do in this course. So please don't. Carrying on with the tools and procedures in biology, and what you're doing in your research project, you're required to gather quantitative data. So when we gather quantitative data, we usually tabulate that data in order to organize it in a table, a data table. We also visualize relationships in the data using graphs. So tables and graphs are very often used, not only in biology but science in general, to eventually be able to make comparisons between groups in an experiment, for example. So in this experiment we have group A, group B, group C, and these are, even though they're labeled plant A, plant B, plant C, they represent multiple plants. If you look at the table over here, plants is plural. So there are multiple experimental subjects in each group, as there should be, making this a more valid experiment. Also note that data analysis includes calculations, so any manipulations of the data, any calculations that are made from the data or with the data, or to transform the data, for example, just to average, that's a calculation, right? So if you're averaging your data, that's part of data analysis. But also description of data. So if you have qualitative data, that would be something that needs to be described verbally. Or you could transform that qualitative data into quantitative data and then place it into a data table. All of that is considered part of data analysis. A lot of you are going to be collecting this kind of data over time in your research project. And what I'm going to be asking you to do is calculate the rate. So whatever change it is that you're measuring over time, that represents a rate. Change over time, that is a rate. So for example, in this case, it's growth over time. And we can talk about a growth rate in this example. And we can talk about the growth rate at different times. That's calculated, and you may recognize this from math class possibly, or another science class. You're calculating the slope of the line. And you can calculate the slope of the entire line, kind of like an average slope. But you can also calculate the slope of any part of the line. And you'll notice that in this example, all three of these groups, the growth is increasing in slope as the plants grow. That means they're increasing in their growth rate as they grow. They're growing faster, so the bigger they get, the faster they grow. That's exponential growth. So whenever you see a curve like this, that represents exponential growth, and these plants are growing exponentially. So the rate, the slope of the line is increasing over time. That means the rate is increasing over time. And you can calculate the rate at any point you just choose a portion of the line and divide the rise. So say we're talking about this portion of the line, we would divide the difference between 10 and 15 here, in other words 5, by the difference in time. And that would give us the rate in centimeters per day, because the measurements here are centimeters and the time is in days. Finally, we would consider this experiment valid as long as everything is correct. And I'll leave you to read this statement, but it just means that the experimenter did everything as they should do in any valid scientific experiment. A huge development in biology was the invention of the microscope. So the microscope is a fantastic tool in biology because cells are microscopic. And we didn't even know that every living thing was composed of cells until we had a microscope to be able to look at every living thing under the microscope to see that it was composed of cells. So there are two different kinds of microscopes in general that we use. The light microscope is what we have here in class to use. And then there's the electron microscope, which costs like $250,000, and we don't have one here at school for that reason. But the electron microscope can magnify much, much more than the light microscope. The light microscope, the limit is about 1,000 times magnification. And in general, in this course, we're not even going to get that high. We're going to get about 400 times in magnification. And the resolution is about 0.2 microns. So that's 1/10 of a millionth. No, I'm sorry, that's 2/10 of a millionth of a meter. So resolution referring to how you're able to discern two dots, how close together two dots are. So if you have two dots under the light microscope, you can tell that they are two separate dots as long as they are at least 2/10 of a millionth of a meter apart. Now that seems like it's very highly magnified, but an electron microscope can magnify 1,000 times more than a light microscope. That means 1,000 thousands, right? 1,000,000 times. So it has much greater magnification and resolution. The reason for that is that electrons have a much smaller wavelength than light, and therefore can resolve things that are much, much, much smaller than light can. In other words, the light, you can think of it as though the light waves will miss things that are too small. The light waves will go right around them. They won't hit them and bounce off of them. So what we're seeing here along the bottom are the same cells under 3 different microscopes. This is the light microscope, and this is about 1,000 times magnification. So these are cocci bacteria, we call them, because they're spherical. They're little round bacterial cells. And in this clump, for example, there are 4 of them. Right here, there are 2 of them that are stuck together. Very similar to what we're seeing in this transmission electron microscope. That's what TEM stands for. This is a coccibacterium that is undergoing binary fission. It's splitting in 2. It's in the process of dividing, cell division. But again, bacterial cells are very simple. That's how they reproduce. They just split in 2. So what we're seeing here is very similar to what we're seeing here in terms of size. So you can see that we can get much greater magnification from an electron microscope than we can from a light microscope. We can also see inside the cells, because in a transmission electron microscope, the electrons are being transmitted through the specimen. Therefore, we can see structures that are inside the cells, as opposed to a scanning electron microscope, SEM, where the electrons are bouncing off the outside of the cells, similar to what you see under a light microscope. The light waves are bouncing off of the outside of the specimen, and that's what is being reflected to your eyes. It's the same thing in the scanning electron microscope. So what we end up with is an image that is 3-dimensional. So we can get a much better idea of what the cells look like. For example, if we were able to shrink ourselves down and actually be in their world, their microscopic world, and stand there and actually look at them. Hopefully, as if you haven't done the cell culture lab yet, you will soon. And cell culture is extremely important in biology. It's simply the ability to grow cells from any source, whether they be bacterial cells or even human cells, in culture, in a dish, in a petri dish, with growth media, and in a particular environment like an incubator. And that allows us to be able to experiment with cells and manipulate cells and learn about cells and observe cells very closely, where we wouldn't be able to otherwise. We can actually take human cells and culture them in a dish. Say we're interested in experimenting with a drug that is going to hopefully cure something in the liver, for example, and so we can culture human liver cells in a dish and apply that medication and see what the results are in terms of the effect on cells. So that's just an example, but it's a very useful tool in biology, so that's why we're looking at it. The final tool that we have to look at here is known as cell fractionation. As you know, hopefully from the hierarchy of biological organizations, cells are made up of organelles, right? So in cell culture, we can deal with a living cell, but if we wanted to see what cells are inside cells and experiment with the components of cells, the organelles of cells, we need to fractionate the cell into its component parts, so to speak. And what we use to do that is a device known as a centrifuge, which is being shown here in the diagram. It's simply a device that spins at very high speeds solutions that contain parts of cells. So we start by homogenizing cells. So we take tissue, which is composed of cells, and blend it. Homogenization is just a fancy word for a blender. We put it in a blender and we blend it. And that breaks open the cells, that releases the organelles into the homogenate here, which is simply a solution that contains all the organelles floating around and cellular components that have been all chopped up by the blender. And then we take those and run it in the centrifuge. In other words, this test tube represents these test tubes that are in the centrifuge. And then you spin it. And the faster you spin it and the longer you spin it, the smaller the components of cells that will be separated out. So if we spin it relatively slowly for a relatively short period of time, then the largest chunks of cells, the largest components of cells, will end up in the pellet at the bottom of the centrifuge tube or the test tube. If we spin it a little bit faster and a little bit longer, then we get even smaller organelles at the bottom of the centrifuge tube. And then finally, if we spin it extremely fast for a much longer time, then that pellet's going to contain the smallest of components of cells, which in this case are the ribosomes, which are the smallest organelles that you'll find making up cells. So it's a very useful tool in separating out the components of cells and studying organelles in general.